Hello and welcome to our educational webinar series, Genome Editing Chats, organized by and for young researchers, supported by Genome in collaboration with CRISPR Medicine News. My name is Laura Torella, and I'm the young researcher and innovator coordinator of Genome Day. Genome Day is our cost action network focused on genome editing for the treatment of human diseases. I'm delighted to welcome you to today's webinar about epigenome and genome editing in CAR T cells. Our speaker for today is Dr. Maria Silvia Azcona. It's my very great pleasure to have you here, Silvia. Thanks for accepting our invitation. Before giving you the chance to present your work, I would like to give a bit of introduction about you to the audience. Dr. Maria Silvia Azcona is a postdoc in Tony Katoman's lab at the Institute for Transfusion Medicine and Gene Therapy in the Medical Center of University of Freiburg in Germany. Her work involves exploring new avenues to mitigate CAR T cell exhaustions by exploring genome and epigenome editing approaches. During her PhD, Silvia established epigenome editing of key monocheckpoints in prostate-specific CAR T. And while in her postdoc, she is currently developing a standardized in vivo exhaustion assay with CD19-directed CAR T cells and exploring it to investigate genome editing strategy that could prevent exhaustion. Silvia is not only an excellent scientist, but is also a wonderful scientific communicator, and you will have a taste of it in a second. I'm super excited to learn more about CAR T cells and what you are doing on epigenome editing. Just to remind the audience, enjoy the webinar and type all your questions in the question box in the screen, and we will discuss all your doubt and question with Silvia in the Q&A session at the end of the webinar. Now over to you, Silvia. You have around 45 minutes. Thanks.
Thank you. Thank you very much, Laura, for this super kind introduction and for the invitation to share my work in this webinar. It's my pleasure to be here. As Laura was already saying, I'm currently in Tony Katoman's lab, but the work I will be sharing today was mostly done when I was in another group, Claudio Mussolino's group, and started during my PhD, as she already mentioned. So let's dive in. I would like to start with an introduction about epigenetic regulation, which we know controls gene expression. And if we zoom into a cell and the nucleus, we start looking at the different organizational levels that the epigenome has. We start by having a 3D chromosome folding, and if we zoom farther in, we can look at the nucleosomes, and within the nucleosomes we have some histones, and that's another layer of control regarding the histone modifications that can make the DNA more or less wound so that the DNA sequence is accessible for transcription. If we look even farther in at the DNA sequence, we can also have another type of control based on DNA methylation, which is normally associated in a way that when there is DNA methylation, the genes are not being expressed, they are being silenced, and vice versa. And this would be what we know as epigenetic control. But I would like to also mention maybe that the newest member of this organizational team, which are the non-coding RNAs, which can offer an extra layer of control for gene regulation, acting as a rheostat of gene expression. So basically, the epigenome tells the genome what to express, where to express it, and when to express it. And it does so in such a sophisticated language, a language that we cannot fluently speak yet, but we do know that some of these epigenetic modifications, which we call epigenetic marks, such as histone modifications and DNA methylation, mostly DNA methylation, can be passed from one cell to the daughter cell. So meaning that it can be inherited. And this makes us think that we can do something called epigenome editing, which would be basically changing these epigenetic marks at our desire. So in our lab in the past years, another PhD student at the time worked on developing a designer epigenome modifier. This is our epigenome editing tool to achieve gene silencing, and it has the architecture that you can observe here. It's basically a multi-domain chimeric or recombinant protein in which we have a DNA binding domain. In our case, in particular, it's a tail-based DNA binding domain, which basically can locate this molecule where we, theoretically, wherever we desire in the genome. And then we have the epigenome, the epigenetic effectors. On one side, we have the strongest mammalian repressor, which is crab. And we know that this transcriptional repressor can induce some silencing marks at the histone levels. And on the other side of this molecule, we have a catalytically active DNA methyltransferase, which can deposit de novo DNA methylation at the desired targets. And it's linked with a sort of a cofactor, which is necessary for the full activity of this enzymatic domain. And we normally try to direct this molecule towards the promoter or enhancer regions so that we can induce the deposition of DNA methylation and thus induce gene silencing. So this is the molecule that we have in our hands, and it's already published that it is possible to use to design this molecule and to target it towards relevant, clinically relevant genes. In this work, they used it to target CCR5 and CXCR4, which are receptors used for the HIV virus in order to infect cells. So this was done in CD4 positive cells, so primary T cells, and they showed that this can induce indeed DNA methylation and histone 3 lysine 9 3 methylation, which is a silencing epigenetic mark, and that it had a remarkable specificity. So just want to make the highlight that this is our tool, but you might have heard of different tools using different DNA binding domains, such as dead Cas9 or zinc finger proteins, and that you can also play around with the effector part. You can change the type of activity by fusing it to TET1 to induce the opposite effect of what I'm showing you here for a gene reactivation, or you can use basically there are lots of options for effector domains that you could choose for these kind of molecules. So since we have so many other tools to control gene expression, I thought it would be worthwhile to mention what are the advantages of having epigenome editing. So we normally think first nowadays we think about the nucleases when we want to control gene expression. And the good thing about the nucleases is that they are very easy to use now with the CRISPR-Cas9 system, and they are effective most of the times. And you only need to design 2 or 3 different guide RNAs, and it's very likely that one of those will act as expected. And they are, let's say, relatively specific depending on the specific guide RNA. But the bad side of this is that it can be highly genotoxic, especially when multiplexing, meaning that when we want to target more than one target at the same time. Another widely known tool is the interference RNA, which can be effective, can be easy to use, can be multiplexable, and also can somehow be relatively specific. But the problem here is that we would require a constitutive expression in order to have a stable effect in the cells. And that means that most likely it's required to have a lentiviral delivery, and therefore constantly modulating or constantly expressing for example, the siRNA. So another potential tool in order to control some protein activity would be the use of antibodies. And those are specific, effective, and multiplexable. You can have more than one at a time. But the problem is that these would have systemic effects. So they would act everywhere where they are available. And that's why we come to our molecule, the designer epigenome modifier, which I will be calling from now onwards DEMS, which is an all-in-one tool. It's a specific, it can be used as a hit and run approach, meaning that one-time treatment would do the job, and it can be multiplexable, given that it would not introduce a DNA break in the genome. The limitations are, of course, that we depend on the CpG abundance. So the CpG denucleotides are required for us to deposit the DNA methylation. The matter about effectivity and stability is it's under evaluation, or it has to be under evaluation for every chosen target and every chosen cell type, because as I said, the epigenome is a highly dynamic system, and we need to be sure that our editing is passed on from one cell to the other. And yeah, so when we have this technology, we thought, why don't we use it to solve a current problem in the field of immunotherapy? And you might have heard of CAR T-cell therapy, and this is basically just engineered T cells to express a chimeric antigen receptor on the cell surface, and this chimeric antigen receptor can now, or is now, providing the T cells with the ability to recognize tumor-associated antigens. And as a consequence of these recognitions, the T cells get activated and they can induce tumor cells death. So about CAR T-cell therapy, we have several CAR T-cell products. They are approved by the FDA and also by the EMA, which is our European agency. And strikingly, they are all against B-cell malignancies, mostly lymphomas, leukemias, and multiple myeloma. And there is a high success rate, but some patients do not respond, and also some patients suffer relapse. And moreover, there are no CAR T cells approved for solid tumors. And what many of us in the field consider a major cause of resistance is a phenomenon called exhaustion, which affects the T cells. So the hypothesis is that the CAR provides the T cells with the ability to recognize the tumor cells more than once. So meaning that the CAR T cells have to be sort of serial killers. And because of this constant and stimulation through the antigen, through the exposition to the antigen, then the CAR T cell becomes overly activated. And this leads to a phenomenon called exhaustion. During this exhausted state, now the CAR T cells and any other T cells that has undergone through a similar problem, they are characterized by a decrease in proliferation, decrease in cytokine production, a decrease in cytotoxicity, and one of the hallmarks is that they overexpress inhibitory receptors. These are also known as immune checkpoints, such as PD-1, LAG3, CTLA-4, TIM3, and many others. So normally these inhibitory receptors are overexpressed at this stage so that they can physiologically control the T cell activation. So what happens in our case now is that these immune checkpoints can now interact with the ligands that are present in the tumor microenvironment, and these can deliver too many inhibitory signals into the T cells. So many years ago, it was discovered that when you block with blocking antibodies, these inhibitory receptors, such as, for example, PD-1 and CTLA-4, which are actually clinically approved for the treatment of cancer, these can prevent the development of exhaustion and to some extent even revert it. And what is interesting about this is that apparently every immune checkpoint acts over different cellular pathways. So when you combine the blocking antibodies against two different immune checkpoints, you can have a sort of synergistic effect. So this made us think that we could use our epigenome editing tool, which is amenable for multiplexing, in order to target at least 2 of these inhibitory receptors. So this is what we thought of doing. And so we thought about editing the CAR T cells epigenome. And currently, for the manufacturing of CAR T cells, the cells have to be retrieved from the patients anyways, and they have to be transduced with either a gamma retrovirus or lentivirus encoding for the CAR transgene. So and then those cells are expanded and then they are placed back into the patient. So we thought our approach would be compatible with this protocol if we take those transduced cells, electroporate them to deliver an mRNA encoding our DEMs targeting PDCD1 and LAG3 separately, so that we can produce epigenetically edited CAR T cells that would have the advantage of not expressing such high levels of PDCD1, of PD1 and LAG3. So I will show you now some of the results we got when we attempted to do this dual silencing in primary T cells. So we basically start with primary T cells with an activation step 3 days before. Then on day 0, we perform an electroporation, which is widely common in the field, to deliver the mRNA encoding for those 2 different DEMs. One targeting PDC1 and the other one targeting LAG3. Then we let the cells recover, they expand in vitro, and then some days later, specifically at day 7, we perform a reactivation step. This reactivation step is crucial in order for us to detect our targets, which are only expressed upon T cell activation, and also to see if this physiological change from non-activated to activated allows the epigenome editing to stay through this challenge. So 2 days later, we perform the detection of our targets, and we kept the cells in culture for almost 40 days and repeated this reactivation step every 10 days. So in total, we perform 4 reactivations, and I'll show you what we get when we do so. So here you can see the PD-1 positive cells fold decrease over time from day 9 to day 39. And you can see what happens when we do single delivery of a DEM targeting PDCD1 or dual delivery in combination with the DEM targeting LAG3. So we get very similar efficiency, either with single or dual treatment. You can see that there is some a slight decrease in the efficiency. We go from 3 to 2-fold decrease, but we consider this modestly stable. Then when we looked at the mRNA levels, because when we are silencing, we are supposed to also see a decrease at the transcript level, because it's the transcription step that is being, let's say, impaired in this treatment. And we could actually see that we have 4 to 2 times less transcript of PDCD1 in either single treatment, which is represented by the letter P here, or dual treatment. And we have no effects over LAG3 in the LAG3 treated sample. So one last thing that we need to conclude that what we are actually doing is epigenome editing was to check if we really do have more methylation at the targeted locus. So we performed bisulfite sequencing of this area of 400 base pairs spanning the target sequence. And what we found was almost 5-fold higher methylation, overall methylation levels at this locus, specifically at the midpoint of this experiment, indicating that there is an association between the DNA methylation and the downregulation of the transcript of PDC1 and also at the protein level. So we did pretty much the same for LAG3. So it's the same experimental setup. And we could also see a stable silencing. So because we found about 2-fold decrease at the LAG3 positive cells percentage for both single and dual treatment. And when we looked at the transcript levels, we again found a decrease towards more or less half of the transcript. And when we wanted to confirm this by bisulfite sequencing, we again found that there was indeed an increase in DNA methylation at this targeted locus. This was all pretty much promising in primary T cells. So we thought, what about the specificity of our treatment? Because before moving on to treating CAR T cells, we wanted to make sure that we were not basically causing an overall impact at the transcriptomic level. So what we did for that as a first step, because this would be a very difficult job, I mean, to establish or to identify potential off-target effects when we do epigenome editing, we basically would need to do whole genome bisulfite sequencing. And then the interpretation of the data, if we find methylation randomly at some available or CpG denucleotides, it would be very difficult to know what does this mean, because the presence of one methylated CpG might not necessarily cause an impact. So we thought the easier way of doing this to begin with would be to look at the transcriptomic data by performing bulk RNA sequencing from samples that were treated, and then looking at the genes that are differentially expressed. And in order to see whether that could have been caused or not by our treatment, we thought we could compare those genes identified with in silico off-target predictions based on sequence similarity of our DAMs. So I can show you here some volcano plots of what we found for the different samples. These was all done in comparison to a mock treated sample. So when we target only PDCD1 or only LAG3, we do not have so many differentially expressed genes. You can see our targets are indeed where they are supposed to be here. So this is another confirmation that our treatment does indeed cause a decrease at the transcript level. And when we look at the sample that received the dual treatment, we strikingly saw many more differentially expressed genes. We do not have yet an answer of why is this happening to our samples. We can hypothesize that since this sample receives double the amount of the DEMS, perhaps there is some unspecific effect caused not by the DNA binding domain, but by the effector domain, for example. Or could also be that having both of them silence is having sort of secondary effects affecting some other pathways. And as I said, we cannot provide an answer to this yet, but I consider this a very interesting open question. So as I said, then we started to look, okay, let's only look at the genes that are differentially downregulated, because those are the ones that are most likely to be caused by an unspecific effect of our DEMS. And then we also thought, let's separate those genes and in order to see those that are consistently downregulated in both samples, single and dual treatment. And when we did that, we ended up with only 2 genes with only 7 genes that were consistently downregulated for the DEM targeting PDCD1 and 8 for LAG3. And then we compared it to our in silico off-target predictions. And coming out from this comparison, for the DEM targeting PDCD1, we only found 2 genes. One of them was our target indeed, and the other one was this gene called TEMIS. And when we looked at the specific target or the potential off-target location, we saw that it was like 20,000 base pairs far away from the starting or the transcription start side of these genes. So we have yet to validate this, but it seems rather very far away as to have an impact in the expression. For LAG3, we basically only found our target, meaning that it's unlikely that those genes were differentially expressed due to an off-target effect. So this, as I said, was one way of starting to have a look at the safety of this tool. And since we were sort of convinced, we moved on to the silencing in CAR T cells. And here we have a very similar protocol. The only difference is that we have to include a transaction day, which is basically 2 days prior to electroporation. And then we let the cells recover. And then we do a co-culture with the target cells, we importantly used a second generation anti-PSMA CAR that was developed here in our lab also. And it's a solid tumor model, and therefore we use this in order to test the functionality of our CAR T cells, and importantly also if our silencing is also stable in these CAR activated cells. So when we do a co-culture, we basically have two ways of performing this. We have an acute setup and also a sort of a chronic setup. And here, when we have this acute setup in which lasts up to 48 hours, we checked whether our CAR T cells that were edited were equally able to be activated in response to seeing the antigen. And you can see here that when we measure CD25 as a proxy of activation, we have basically undistinguishable activation amongst our different samples, meaning that our treatment does not impair the capacity of these CAR T cells to become activated. And very importantly, also, when we looked at the PD-1 positive CAR positive cells and LAG3 positive CAR positive cells, we did see that our treated samples had decreased levels of PD-1 and LAG3, both for single treatment and also dual treatment. So this confirmed that our editing can also work in this protocol with CAR T cells. And so for an acute setting, that was also, again, convincing for us, but we wanted to know whether the epigenome editing would last over a long period of activation. So for that, we did this 12-day co-culture, which entails adding more target cells every third day. So this would be a repeated antigen encounter setup. And at the end point, we took those cells and looked at the methylation levels to see whether the methylation marks are still present at this point. And indeed, that's what we could see. You can see this nicely also in this heat map, where the samples that were treated were more or highly methylated in comparison to the control. The same, so this was for PDCD1, and pretty much the same picture was seen when we looked at the LAG3 locus, so indicating us that our epigenome editing can be sustained over this long period of time with serial activation of the CAR T cells. The last thing that we wanted to do was, of course, to see some functional data. And for this, we performed some in vitro assessments. We started with the most basic question, which is, can those CAR T cells still kill their target cells after this treatment? And you can see here that indeed there was almost no difference in the killing capacity of the treated samples in comparison to the untreated sample. The same goes for the secretion of inflammatory cytokines like interferon gamma and TNF. Then when we looked at the T cell subtypes that are present in our samples, we found, again, almost or basically no differences, neither prior antigen encounter or post-antigen encounter, meaning that it's not like our treatment is causing those cells to be more terminally differentiated, or so. We retain basically the same distribution of T cell subtypes, which is another indication that our treatment is safe. Then when we looked at the proliferation over repeated antigen encounters, again, we found basically very similar behaviors among all the different samples. So coming back to the topic of T cell exhaustion, as I said, is one of the big challenges for CAR T cell therapy, and also to continue our efforts in order to produce CAR T cells that are resistant to exhaustion. And because we were working previously with this solid tumor model, and I showed you all the nice data, but we tried for a long time to develop a setup where we could actually see a benefit from our silencing. And this involved transducing those target cells with lentivirus encoding for PDL1, which is a ligand for PD1, but basically we could not see an improvement in the functionality in those assays. And also we could never really prove that the repeated antigen encounter model was actually leading to exhaustion. For some reason, working with those cells was somehow technically very difficult to induce exhaustion. We try different culturing conditions, we try hypoxic medium, we try adding the ligands as a recombinant protein in the supernatant. And yeah, basically we did not manage to achieve exhaustion. And then we moved to a different CAR T cell model, which is the CD19 CAR T cell model, very well known in the community as the first CAR T cell therapy that was approved. And what we did in this second project that I want to mention is we try to develop an in vitro exhaustion assay so that we can explore genome editing strategies that could prevent this. And that's what I want to show you now towards the end of the webinar. And what we do basically is we take those CD19 CAR T cells and we perform an enrichment so that we have only CAR positive cells. This is done with max selection. And then we start a long protocol of at least 10 to 14 days, in which every second day we add the target cells, which in this case are NAM6, and they are expressed in GFP so that we can track them over time. And every second day we add more NAM6 in order to keep a fixed ratio of one CAR T cell per 4 NAM6. So a 1:4 effector to target ratio. So this is a very labor intensive protocol because it requires periodic attention, and every second day we have to count the cells, stain them to check for the CAR, and then basically add more NAM6 so that the experiment continues. And by the end point, we sort the GFP negative cells, which are the original CAR T cells, and we perform some endpoint analysis. So over time, what we can see is that when we take different donors for T cells, healthy T cell donors to begin the experiment with, we have a period of expansion at the beginning. So the cells, the CAR T cells are expanding in response to the antigen, and then there comes like a peak, and the proliferation starts decreasing. So this is measured just by the absolute CAR positive cells, but you can see that it's the case for all the 3 donors. They have different kinetics and it's extremely variable. But it does seem to be consistent in the fact that towards the end of the assay, the number of cells is decreasing. And when we measure some exhaustion markers and some stemness markers, what we see, in this case, I'm showing you only CD39 as an exhaustion marker, and IL-R7, which is receptor for IL-7, we can see that they match what one would expect when one wants to induce exhaustion. So CD39 is increasing over time, and IL-7 receptor is decreasing over time, indicating that the cells are more exhausted and they have less stemness. At the end point, as I said, we take those cells and we perform one last co-culture at a smaller scale where we can measure the cytotoxicity properly. And when we compare cells that from this endpoint with cells that were just left in culture untreated, what we can see is that for all the 3 donors that we have tested in this set of experiments, we can see less cytotoxicity in the cells that underwent this repeated antigen encounter in these conditions, meaning that those cells are presumably exhausted. And again, the same happens for when we check for example, the secretion of IL-2. We could not detect any IL-2 in the cells that underwent this treatment, but we could do so in the controls that were left untreated. So for us, it was sort of convincing that we finally had a model that can resemble exhaustion. And the last thing that I want to show you is that, as I said at the beginning, the goal is to test genome editing strategies that can prevent exhaustion. So we thought, why don't we include a control that is very famous to promote proliferation of CAR T cells and promote, let's say, the opposite of exhaustion. So we took this target, which is TET2. There is a very famous case of a patient who had a striking recovery from his condition after receiving CAR T cells, and this was all because of a mutation in the TET2 locus. So yeah, we did the same basically, but in vitro. We induced indels using CRISPR-Cas9 at the TET2 locus, and then we took those cells and performed the experiment I just mentioned before, the exhaustion assay. And basically, you can see here in purple is the TET2 knockout. We have very high variability between different, among or across different donors, but the tendency is that those cells that have TET2 knockout proliferate better, and those are also, those cells are expressing higher levels of an stemness marker. This is another stemness marker, TCF7, which encodes for TCF1. And this goes along with what was reported also in the literature about TET2 knockout samples. We also have better cytotoxicity and a tendency of higher secretion of, for example, interferon gamma. So this was a nice confirmation that we have an assay now in our hands that we can explore or exploit to test different strategies to rescue those cells from exhaustion. And this is what we are doing now. Unfortunately, I cannot really tell you what we are testing because this is it's non-disclosable data, but I thought it would be nice to share this sort of teaser of what we're working on in terms of preventing exhaustion in CAR T cells using epigenome and genome editing tools. So we have reached the summary, and I would like to conclude by saying that epigenome editing is a novel technology that allows precise modulation of gene expression, as we have seen for the 2 targets that we chose. And for us, to our knowledge, this is a unique report of multiplex epigenome editing targeting 2 highly dynamic genes in primary cells with high specificity. I say highly dynamic because PD-1 and LAG3 are genes that are overexpressed upon activation, and then when the cells are in rest state, their expression goes back down, and then if we trigger again the activation, then the expression goes again up. And for us, the fact that we could retain the DNA methylation marks and the silencing throughout these challenging experiments, it means that the silencing is stable. And this is compatible with the CAR T cell technology that we have nowadays, which involves the delivery of the CAR with a lenti or a retrovirus. And it can also be done in a hit and run approach. And it can also totally be applicable to other models. And the induced silencing persists through replication and physiological challenges, which we have which have done by repetitive activation, and importantly, does not impair functionality in vitro, at least in all the tests we have done so far. And also, lastly, an in vitro exhaustion assay that we have developed is amenable to test gene editing strategies. So this is my acknowledgement slide. As I said before, this work was done when I was working for my PhD and also my first postdoc in Claudio Mussolino's lab. And I have highlighted here the people that have contributed to the work I presented today. And I'm looking forward to your questions. Thank you.
Thank you, Silvia. This was brilliant. I wasn't expecting anything less from you. And it was just so clear and so educational. Thanks, because you achieved completely the goal. This was my question, making your presentation educational for our young audience. And this was absolutely great.
Thank
And
you.
thanks for starting with a very nice introduction on differences between your approach, epigenome approach, and different approaches that has been used until now. And then, of course, for the overview on your approaches. And it was amazing listening to you and speculating about hypotheses why your approaches didn't work and what could have been done to modulate and make it better. So thanks a lot. This is not so usual, so thank you so much. While we wait for some question to came up from the audience, and I'll remind everybody that we have our Q&A box. So type in the question. I do have a few, and maybe some that can help also audience that is not so familiar with CAR T cells. And maybe if you can speculate a bit more on TET2 and why, what is TET2 and why having these knockouts help your approach.
Yeah. So TET2 is sort of a master or a key member of the epigenetic regulation, because what it does, it basically starts the demethylation process of the DNA. So these molecules, the DNA methyltransferases that participate in this in the deposition or removal of DNA methylation marks, they are basically controlling the accessibility to these genes for transcription. So the case of TET2 is particularly interesting because of what we have seen in this patient. And this patient ended up having a dual mutation in both loci of TET2, and ended up having this striking recovery and having basically a monoclonal population of CAR T cells. So it was one cell carrying this mutation, and then it expanded so that the patient had this amazing recovery. And ever since, people are trying to understand why is this molecule so interesting or so important to promote T cell proliferation and sort of maintenance in this stemness state or in this stem-like state, but there is still no clear answer. It seems to be involved at very different stages of T cell differentiation, and so we do not have yet. I could not tell you yet exactly why do we see this, and because another interesting thing is that we have seen this patient recover with this mutation, but we also do see TET2 mutations in patients with T cell lymphomas. So it seems that it somehow it's like a driver mutation for the development of T cell malignancies also. And that's why we ourselves do not want to use TET2 knockout for like a clinical trial and put those cells purposely in patients because of these precedents of T cell lymphomas, and which is a risk anyways for CAR T cell therapy. So that's why we want to use it just as a positive control to see if we can rescue exhaustion, but now we are focused on different targets.
Awesome. Thanks a lot for the clarification. It was actually another question, coming up question. Do you have other potential target now? And have you already considered others target instead of that too?
Yeah, exactly. So we actually have sent those samples from our exhaustion assay for RNA sequencing in order to identify potential new targets. As I said, I cannot disclose them, but we are we are trying to find something novel and potentially helpful.
Looking forward to the publication then.
Oh, yeah.
Well, we do have a lot of questions coming up from the audience now.
Cool.
I will start with one question from Roland Letters. Sorry if I mispronounce your name. I said this year we'll see the first clinical trials establishing CAR T cells in vivo. No ex vivo expansion using LMPs. Do you think that exhaustion will still be an issue? As I believe and I read that there's some indication that exhaustion may also be linked to the degree of ex vivo expansion.
I think it would be very interesting to see. I personally think that it will remain as a problem, even though the delivery of the CAR would eventually be in vivo or extracorporeal and then introduced back. So I've seen recently that it's a different avenue. But even so, I think exhaustion will remain a problem because it's something coming from the tumor microenvironment, which is still there. And I think we would need to combine it with a way of modulating the tumor microenvironment, maybe secreting some cytokines or to make it sure. I think it basically will continue to be a challenge because of the signaling that are driving the signals that are driving exhaustion.
Yeah. Maybe also one of the reasons why you haven't seen exhaustion in the first part of your projects, right?
Yes.
Also, I was wondering, and this is completely naive question, but if you have considered other cancer microenvironments, and so maybe the one you choose and the specific one you choose and the silencing of the check-in that you chose wasn't good enough or high enough to fight with the microenvironment, and that's why you haven't seen.
Yeah, so we basically do not see, like, if we take this assay and we try to establish to begin with a positive control, which would just be transduced cells and normally expressing those targets, and we try to expose them to the ligands by either by overexpressing it in the target cells or by adding it to the supernatant as a recombinant protein. We see basically no impact in the CAR T cell functionality. So I think this is something either depending on the CAR, because there is a recent publication from Sonia Guedan's lab in Barcelona where they actually showed that the CAR affinity, it's a predisposer. So if the CAR affinity is too high, then the PD-1, PD-L1 axis has no room for modulation of the CAR T cell activity. So I believe our CAR is very high, has very high activity. We do have some data about that from the SCFB that we took. And that's one of the potential explanations why we didn't see any effect.
Yeah, thanks. Then we have some other questions. So Francois Bilco asks, is there a risk for double-strand breaks with them? If so, did you evaluate the risk of inducing structural variation with your DAM? And a next question, what would induce a risk of oncogenic transformation on CAR T cells?
So theoretically speaking, there should not be DNA cutting activity in these molecules because there is no nuclease capacity. We did not test this. I mean, we could maybe set up a translocation PCR to see if PD-1 and LAG3 are somehow translocated after our treatment. I do believe there is some data from other people doing epigenome editing showing that there is no cutting, but because of the principle or the knowledge that we have about the activity of this molecule, there should not be the DNA double strand break. But yeah, I cannot tell you 100% no because I haven't done it. But yeah, this it's a good question. Yeah.
Thanks a lot. I also loved a lot your approach on RNA-seq bulk. And actually, one of the questions that came out was also, what's THEMIS? And what's the relation of THEMIS with PD-CD when?
No relationship, no.
Okay.
I mean, yeah. So it is complete, like it's in a different chromosome, and the supposedly off-target location is so far away from the transcription start site that we do have to validate this eventually, of course, with to really check if there are some methylation marks changed at this place. But whether this will have an effect or not on THEMIS, we also have to check it by qPCR, maybe. Yeah.
Then we have a question from Kai. Hello, Dr. Silvia. In CRISPR-Cas technology, the PAM is used to target the gene of interest. How does this work in epigenome editing and when there is no cutting or insertion or new genes?
Yeah, so as I said before, there should not be cutting, but we do not require a PAM for the tail. The tail basically can bind any theoretical sequence because they are made from modules that you can choose, and each module, it's let's say each nucleotide has a specific module that you need to assemble in that order, and the only sort of condition that we have is that the first nucleotide has to be a T, just because in that way the structure of the tail is more stable and is supposed to bind better. So this is the only constraint that we have.
Okay, thanks. And then we have a question from Andrea Lopez Marreiro. Great talk. Based on all the positive data you presented, what else would be missing to test this treatment in vivo? And do you have an idea of what animal model would be preferable? And thanks in advance.
Okay, so a lot of things I would say. I would say first we need to figure out what's happening with all those differentially expressed genes that we have in the dual treatment. As I mentioned during the presentation, that would be for me something very interesting to know and to be sure that we have at hand a safe technology. And the other big question remaining, and why it is really hard for us to publish this story, is that we are currently in revision, but everybody keeps on asking for functional data, and we cannot show that our treatment makes the CAR T cells better. So we need a model for this. Absolutely, we need to test this. As I said, it's in vitro. It has been difficult so far. I would imagine that a humanized mice model might help. Where we would have cells that are expressing those ligands and perhaps somehow mimic the tumor microenvironment there. So it's a complicated setup because of the need of we need the ligands to be present, and we need to sort of have different cell types. And yeah, therefore, my only thought so far is to have a humanized model and to see if our treatment actually makes a difference. I mean, we would expect so because of the vast evidence in the literature where people are reporting this, and because having these targets blocked with monoclonal antibodies is, I mean, it won the Nobel Prize, I think in 2018. So it's nice to think that it would work, but I think we definitely need to prove this before we bring it for farther.
Yeah, and what the humanized model recapitulates the microenvironments of the tumor?
I don't know of any actually. I think we would have to establish something for that, or a search for a collaboration or so. So if somebody's out there interested, I would be very happy to collaborate.
Sure, thanks. And we have one more question, which is from Alfredo. Have you already tested if there is partial reversibility in methylation induced by the transient expression of the mRNA after some time, for example, weeks after editing?
So we have done the two time points the bisulfite sequencing to check for the methylation in two different experiments. So once was midway of the culturing in the T cells, in the primary T cells, and one was endpoint in the CAR T cells. We do see that the methylation remains there, but we don't have like a sequential analysis because we assume that there is this association. It would be interesting to know if we lose something over time. I mean, we do see that LAG3, for example, remains like at the same level of silencing throughout the experiment, which for me would indicate that the methylation is consistent. But as I said, we do not have these data. And the reason that we don't have it is that we think it's not necessary once we have shown that there is this association between the expression levels and the DNA methylation. But for PD-1, for example, we do lose a little bit of the strength of the silencing over the time in culture. And perhaps it is because we lose some of this methylation, because this is possible. This is why we put so much effort into checking that this is stable over time. As a proxy, we use just PD-1 expression as a protein, and but yeah, we would have to do that, particularly focus on that question in order to answer this.
And then will this affect the persistence of your CAR T cells?
I would say so. If it is somehow reverted by a mechanism that's called replication dependent dilution, for example, which basically means that during copying these to the new cells, then one mark is lost. So it's like an error that the cells make. So this is totally possible, and this would of course affect our intended editing or the efficiency of these cells.
Okay, and I think we have time for one more question, and this question comes from Roland's letters. I think was the same that asked in the beginning. So yes, a lot of questions. So for the modeling question, you may consider 3D culture humans tumoroids derived from human primary tumors. That's what we use in-house to test CAR T cells functionality. So I think he is also suggesting potential
that's an amazing suggestion. Yeah, it's an amazing suggestion. It would be nice if we could at some point get some patient-derived cells and try to do this. This would be actually a very nice in vitro model.
Yeah,
Thank you.
I think you should establish a collaboration with Rollins.
Yeah, drop me an email.
Sure, thanks, Roland. So thanks a lot, Silvia. Thanks for your time. We had an amazing time together. I think we have no more question coming at the moment. But happy to send you all the questions we will receive afterwards, in case there are any. I received very good feedback also from CRISPR Medicine News. They are super happy with your webinar and how it works amazingly. So with now, I will happy to end this presentation. I remind the audience that we have a records of this webinar, and this will be available in pretty soon. And you have time, in case you haven't, to go through the entire presentation of Silvia and eventually send me emails with your additional questions. And to the audience, I want to remind you that next month we do have another webinar. And the invited speaker for next month is Dr. Nerea Zabaleta. She's from the Center for Applied Medical Research, CIMA, at the University of Navarra, Spain. And we're going to change a bit the topic. This time we're going to go for viral delivery for in vivo editing. And hope to see you very soon for the next webinar. And for the rest of you that won't see at the webinar, at least see you at CRISPR Mass in Copenhagen. So thank you very much, Silvia, again, and see you very soon. Bye.
Bye.